we had been talking about naming these aldehydes and ketones. And so last time we had structures, we did the name. Now we're given the name and we can do the structures. And so aldehyde is going to end in al, or aldehyde. Ketone is going to end in -one. So when we see hexanone, we know that's a ketone. And my recommendation would be to just, when you have S and R, just draw it, and then do the ranking. And if it's right, good. If it's wrong, just switch it the other direction.
Okay, yeah.
And it's quicker that way. But I mean, of course, it's up to you, whatever you're.
It's like draw it without the stereochemistry, and then just kind of
draw it with stereochemistry.
okay.
Just pick one,
Pick
right?
one,
And
okay.
just see if it works.
Okay, yeah.
Rather than try to say, okay, now which, like.
Yeah.
That's the way I do it. So
Cool.
So again, and the way I also do it, I like to pick out the parent name and start from that, right? So cyclohexanone, that's going to be essentially a hexagon with a ketone on there, right?
Yeah.
And then that's going to be 1. So 3,3-dibromo, so that's going to be 1, 2, 3. So bromine there. 4-ethyl, right? So that's going to be our S.
Okay.
Okay.
Yeah.
And depending how it comes off of that, we could make it a wedge, and then rank it from there. Okay. And see if that's S. So here,
Towards the bromo's 1.
bromo's 1. The ring is going to be 2 because it's got a longer group. 3, so it's, and that's
S.
S.
Cool.
So
Right.
I like to do wedges first because then I don't have to then superimpose it with the hydrogen. If that was a dash, then I would have to rank it and then flip it. So, but whatever you prefer.
But like, if I drew a wedge and then was like, oh, it needs to be dashed, and like drew dashes through it, would that be like apparent, or better just to have a pencil and eraser?
Probably, because if you did this, I'm like, well,
Yeah, it's kind of both.
is it a dash or is it a wedge?
Yeah, okay, fair.
Right?
Yeah.
So probably best to erase it.
Yeah, cool.
Or, I mean, if you said should be a dash,
Okay.
I would accept.
Okay.
Okay, pentanone. So not cyclo, right? 1, 2, 3, 4, 5. 1, 2, 3. So the
3.
ketone is on the 3rd, and then 2, 4 dimethyl. So there's a methyl here and a methyl there.
Cool.
1, 2, 3, 4. Yeah.
Awesome.
That's it, right?
Yep, that's what I got.
No stereochemistry, so we're good.
Okay.
All right.
3.
R3 bromobutanol. So bute 4. 1, 2, 3, 4. And there's no number with the bute now, so I guess we can assume it's at the first carbon, right? Because it's al, so that's going to be an aldehyde, so that'll be a hydrogen.
Okay.
3 bromo, 1, 2, 3. And again, you can do your dash or your wedge, or you can just, before you draw that, you could just put a placeholder there and do it. It doesn't really matter.
Okay.
But what's going to get the 1? It's going to be the one closest to the ketone, right? And then
2 would be the rest of the alkane chain. Alkane, yeah. And then
3. So that's R.
Oh wait, actually, would bromo would be number 2, right?
Oh, because it's bromine.
Yeah.
So I messed you up there.
No, I just
I forgot to put the
Yeah.
I forgot to put the bromo on there.
Okay.
So that would definitely make that 2. 3.
Would it be number 1 since it's
Or
really close to?
Actually,
yeah.
it's
Sorry, I'm
Yeah.
still waking up too.
That would be bromine's a halogen.
Mm-hmm.
Super electronegative. That's bromine's gonna be 1. Then it's 2
Yeah.
for the ketone. Then it's 3.
And that would be
And then that's-
R.
That's R.
Okay, cool.
And that's what we want, right?
Yeah,
Okay.
cool.
All right, so just a couple slides on this slide deck, and then we can go to the next one. Oh, shoot. I didn't. Let me print one off for you. Let me print that. Oh, this is printing.
Yeah, I kept messing up the RS like early in the sequence. I can hear Sean's voice still now. Like, that helped. Though.
Good.
Yeah.
That's good. That's what you need,
Yep,
right?
Yep.
Yeah, bromine. So obviously it helps to write the structure correctly. If you forget something like I did, then that's going to change it up. But,
Yeah, if you forget to add that halogen on there, yeah, it's
Yeah.
easy to.
All right, so how do we make aldehydes? There are several different ways to do it, right? Using different reagents and giving us different products in the end, or allowing us to use different starting material, okay? So if we have an alcohol, and that alcohol is a primary alcohol, it just has one other carbon group, we can do one of 2 methods. And what that will do is, that will essentially rip off that hydrogen there, okay? And so we end up with our ketone. We have to use these reagents that are strong oxidizers, okay? And so that's what helps facilitate that.
Okay.
Now, if we wanted to use ozonolysis, which I think you went over that last quarter.
Yeah.
That sound right?
Yeah.
Ozonolysis.
Oh, yeah, it cuts the pi bonds, yeah.
Right? And then hydroboration oxidation. I
Yeah.
did that last quarter too.
Yeah, non-Markov.
Yeah, so we have that boron complex, right?
Yep.
That's anti-Markovnikov.
Okay.
Right?
Yeah.
So if we want to take an alkene as our starting product, then we can use ozonolysis and end up with 2 ketones.
If you had
Or aldehydes, I mean, sorry.
if the hydrogens were replaced with carbons, would it make a ketone
Exactly.
the same way?
So
Okay.
you're one step ahead.
Oh, I'm sorry.
Yeah. No,
Okay.
no problem. No, I like it.
Okay.
Like the way I like it exactly here.
Okay.
Right? So
So it's just whatever you start with. Yeah.
yep. So we can use that same reaction just with secondary, or I guess no, tertiary carbons on those alkenes here.
Yeah.
Those alkene carbons to make the ketone instead of the aldehyde. And then if we take an alkyne and go through the hydroboration, then it allows us to put this oxygen here. So it's not Markovnikov, right? Because if it were Markovnikov, it'd put it here, okay? Because there's a hydrogen
Hydrogens
here,
go with the
so
rest, yeah.
so the hydrogen's going to want to go there. So if we did want to make a product like this, hydroboration oxidation would be to do it.
Is that because like boring's really big, it gets in the way, so it forces it to go the other way?
Yeah, if you remember, it forms that big kind of ring, complex intermediate,
Okay, right.
and then it kind of protects it
Yeah.
so that it can't tack it on here. It
Okay.
has to- that's the only place where it can go.
Yeah, it's coming back. Yeah.
Now, if we wanted to then make ketones, as you astutely predicted, we just switch our starting material and do an ozonolysis, or we take secondary alcohols, right? So primary alcohols for aldehyde, secondary alcohols, and we use sodium dichromate. Instead of going back here, we use that pyridinium chlorochromate, or, oh gosh, DMP.
Dimethyl,
No, DMS, so DMSO is dimethyl sulfoxide.
Sulfoxide, yeah.
This is Des Martin something or other.
Oh, yeah.
Anyway, not gonna make you memorize what those acronyms are, but so we use different tools, right? And we use different starting material, but we're still oxidizing it, and that's what's gonna be the difference between an aldehyde and a ketone.
Is there a way, like looking at the formula, to tell it's an oxidizer? Is that just something you kind of like remember?
Not with this. I mean, I wouldn't know of any good way to tell if that's an oxidizer.
Yeah.
DMSO is, and here this is like an alkyl halide, but there's no great way to know that.
Yeah.
That I'm aware of. If there is, then it's lost to me, unfortunately. So basically it's all the same with the exception of Friedel Crafts, right? So oxidation, ozonolysis, acid catalyzed instead of hydroboration, but we can still use that alkyne, all right?
Yeah.
And it's going to be Markovnikov in this case, because we're not doing the hydroboration step. We are taking the alkyne, we're treating it with something different, sulfuric acid, water, mercury, and so then we make our ketone that way. And then last but not least, if we had a benzene ring, right?
Yeah.
You remember from Friedel Craft, we just pull off that halogen and stick on whatever that halogen is connected to,
Okay.
right? So,
So you kind
that
of
would be a way to facilitate that.
you have to start with like sort of a aldehyde to make that benzene ring and an aldehyde product.
We can start with a ketone.
The R. I'm sorry.
Yeah.
We're seeing that as a hydrogen. Yeah. Yep. Yeah. Yeah.
Yeah, so
Okay.
you would start with a ketone
Ketone,
of some sort. Well, technically, that's not really a ketone because a ketone has R groups on either side.
Okay.
So, but you would have the makings of a ketone.
Yeah.
So it's not a true ketone, but you pop that off and stick it on there, and once you do, now you have the
I see.
R group on either side.
Can you use Friedel Crafts for to make a aldehyde, or would it not be stable?
I don't see why not. If this were an H, I'd have to look back at Friedel Crafts and see, is there ever a case? Because usually Friedel Craft has a long R, has some kind of R group on it. But I don't see why not. But I don't know why they didn't include that
Yeah.
in preparation of aldehydes. Because this is the last slide in this deck. Here is Friedel Craft, but it's yielding a ketone.
Yeah.
We'll have to look into that.
Okay.
Yeah, that's a good question. I don't know. But let's try this. So if you want to keep out your preparation cheat sheets, right?
Yeah.
Let's see, how would we get from, what is this? This is a secondary alcohol.
Secondary,
right?
Ketone.
To a ketone. Let me just look back here. We use sodium dichromate.
Okay.
And in the presence of sulfuric acid and water to essentially take off that hydrogen.
Okay. And then it gets the other hydrogen probably from the water or something.
Yep.
Okay.
And then B, so what are we doing here? Here we're using a primary alcohol, so not sodium dichromate, it's that one where we use PCC or DMP or DMSO, followed up with, this is 3 ethanols, or 3 ethyl groups attached to a nitrogen.
Okay. So you kind of need like specialized reagents to do that on the primary.
Yes, and that's why, again, this goes into that. I don't know if Sean, when I taught the whole series last time, I told students, you know, I want you to build a toolbox, right?
Yeah.
All of these things go into your toolbox. And so it's an imaginary toolbox, but like as you're working on your motorcycle, right?
Yeah.
Certain parts require certain tools, and I'm sure if you've ever tried to do a job without the right tool,
Yeah.
and I was going to make this work, it doesn't work as well, right?
No, not at all.
So I like to think of these synthesis problems as tools, right?
Okay.
And so not wrenches and things, but reagents, and so again, my philosophy is not to have you memorize all of this stuff, but hopefully, you know, if you were to have problems like this, you know, you'd say, oh, okay, well, let me see my toolbox, right?
Yeah.
So yeah, it is specialized.
Okay.
Very much so. So DMP we use here, PCC. Here we have an ethyne, and we're making anti-Markovnikov aldehyde, so that's the hydroboration, oxidation. And so to do that, right, we would use these reagents.
The hydroboration, yeah.
C, again, we have our ethyne, and
that one is Markovnikov.
it's going to be Markovnikov. We're doing a ketone, so that tool is going to be here, the acid catalyzed reaction. D.
Yeah, that was.
I already did that one. Okay, E. So, what are we making? Ooh, we're making a diol.
Yeah.
Oh, no, it's not a diol.
No,
diol would be 2 alcohols, but it has an aldehyde and a ketone. So that's a little bit tricky. I actually had to look this up. Like, how would you name that? Because remember we were talking about, you know, ranking and order and stuff.
Right.
And it's a weird one. I had to write it. I wrote it somewhere.
It's the ketone and an aldehyde.
Oh yeah, it's 5-oxohexanal. So hexanal, the aldehyde takes preference over the ketone.
Interesting.
So we name it 1, 2, 3, 4, 5. 5-oxohexanal. Which I wouldn't give you a problem like that to me.
Yeah.
But just for FYI.
Interesting.
And so if we look back,
Ozonolysis.
right? Ozonolysis, yeah. That's what we would do for that, because we can break that apart, but we can also have it connected together, right? Ozonolysis there. So, last but not least, we have
Friedel-Crafts,
benzene, Friedel-Crafts, yeah. So we would just start with like that.
Okay.
Right?
Yep.
And then we'll use our aluminum trichloride. Or if this was bromine, we use our bromine.
Oh, right. It's a different metal. Yeah, iron. Yeah.
Tribromide.
Yeah.
All right. So that's it for that. Any questions on that one, Rob?
No, thanks. It's good clarification.
Okay.
It's cool to see the old reactions come back a little bit.
Yeah,
Not
right?
just brand new ones. Yeah.
They aren't going away.
Yeah.
So now that's how we make those things. Let's talk about specifically the reactions that take place. So we'll get into nucleophilic addition, so a nucleophile attacking that carbonyl carbon. All right, so we end up putting a hydrogen, or if it's, well, it depends on the step, right? But if we start with an alcohol, we can take that hydrogen off of that alcohol group, turn it into a carbonyl group, and then the nucleophile comes in. And then specifically, we can use different types. We can use oxygen nucleophiles, nitrogen nucleophiles. We can even use halogens as nucleophiles, even though they're electrophilic in nature. We can kind of direct them in an acidic environment to be more of a nucleophile than a leaving group.
Okay.
Sulfur, so there are multiple different approaches we can take. We'll get into these in detail, not so much the other ones. Okay, because those are the major ones that are more commonly used. All right, so here we go. We're starting with a carbonyl in this case, and we have our nucleophile, and that nucleophile is going to come underneath. Okay. Oh, let me print that off.
Oh, cool.
I did print it off. Let me go get it for you.
Thank you.
All right.
Thank you.
You're welcome. So, we have the nucleophile come underneath, and then we form this intermediate. It's called an alkoxy group. So this thing right here, alkoxy. Okay, an oxygen with this negative charge. And then we're going to do a proton transfer. It's going to pull off that hydrogen, turn this into an alcohol. Okay. All right. And if you notice here, there's going to be some partial positive charge. That carbonyl carbon is going to be partially positive.
So it's a target. That's
So it becomes a target, right? And it turns out, it's not on this slide, but let's see if it's, yeah, so this slide. It turns out that ketones are going to be less reactive than aldehydes because the ketones have two R groups on either side to help stabilize that partial positive, whereas the aldehyde only has one.
Yeah.
So it doesn't get as stabilized, so it becomes a bigger target, or it facilitates being a target more easily for the nucleophiles.
Okay.
Okay. So that said, both of them are going to react. It's not going to be it's not that much more challenging for a ketone. It just means that our approach is going to be a little bit different. It's the toolbox, it's going to be a little bit different. Okay. So here are a range of some of the nucleophiles we could use. Oxygen, we're going to talk about that a lot. Sulfur, we're going to kind of gloss over and not really go into the mechanism. So I'd like to just focus on the mechanism of oxygen, mechanism of nitrogen. But keep in mind, we could use hydrogen. This is did you guys ever talk about Grignard reactions?
Yeah, Grignards,
okay.
yep.
So we can use, we're actually going to do a Grignard reaction next week. We're going to prepare our Grignard reagent, and then hopefully should be okay for next week.
Okay.
in order to make Grignard reagents, you can't have any water present.
Oh, okay. I remember Sean mentioned that.
Which is nice in tile because it's pretty darn dry. You know, if
Yeah.
we were trying to do this in Florida,
Mhm.
it'd be a little bit. We would need some more equipment that prevents that. But, so a number of things we can use for this, and what would this look like? So how do we figure this out? Well, we have our in this case, we're gonna have a ketone, right? And we have it in the presence of an acid. That acid is gonna deprotonate, and we get a proton transfer to that oxygen. And from there, the nucleophile comes in and attacks, right? So by weakening that carbonyl, like you said, it makes it this easier target for the nucleophile to come in, right? And so it's essentially like the opposite of creating a good leaving group,
Okay.
right? We can facilitate a good leaving group to leave. In this case, we're making the carbon a good nucleophile for something to come in for an addition reaction. Okay, so that we're taking a ketone, turning it into an alcohol, okay, or something different depending on what that nucleophile is.
Okay. Yeah.
And we'll get into more specifics here. So, what would this look like? What could we use? Well, here we have a Grignard reagent, and we're starting off with a ketone. And so, that last slide we talked about how to make ketones. This slide deck we're talking about how to take ketones and aldehydes and react them to make some other product.
Kind of undo
So
it.
it's kind of the reverse, right? But we can use a Grignard reagent. And let's see, where's my marker? How would this look? We have the Grignard reagent come in. So we have our ketone. What is that? That's a cyclohexanone. And we have what's it? An ethyl. Okay. That's going to pop in down here, right? And then we'll put in some water. And the water is going to then protonate that oxygen, right? Because
So we have like
steal
charge.
that bond there, right? So we'll end up with here and then ethyl. And so now we have, drew this out as water. And then for this one down here, right? So we have
Acid.
the acid, and so now again, right? What's on the
Can like protonate the oxygen.
so right, and that's gonna
Okay, and then you get that carbocation on the ring, and then so that the chlorine attacks that. Okay.
So the chlorine in this case becomes a
Nucleophile.
nucleophile, right? And that's kind of the thing where halogens, we've always talked about in the concept of being good leaving groups, right?
Yeah.
But if we have an acidic environment,
Okay.
we can essentially coax that to be easier to attack and become the nucleophile.
Okay.
So, so we have this double bond. Double bond here. Now the chlorine, right, is still there because it just lost its hydrogen, and it's deprotonating anyway. So like it's not. It could be that the oxygen takes that off of that chlorine, but we know that hydrochloric acid is a very strong acid, and it may just be giving off hydrogen protons in that solution.
They're disassociated in the solution.
Dissociated, yeah.
Yeah. So basically, yeah, the acidic environment like kind of favors breaking apart the ketone or the aldehyde, that double bond there.
Yeah. So this will come in here, right? And this will go here, and then we actually, at that point, have our product there.
Okay.
So, kind of heavy on the mechanisms today,
Yeah.
which will hopefully help with the mechanisms on that practice exam.
Yeah. Yeah, usually it's like a new reaction. I don't like understand right away, but once I like dive through the mechanism and work through it, it like helps like make sense. And
And the nice thing is that some of these are hopefully not super new in terms of their reagents and stuff. Like Grignard, you've seen that before. Hydroboration, ozonolysis. But yeah, I get that. Sometimes you're just looking, okay, well, what's the reaction itself? And then the mechanism is just, okay.
Yeah.
So you kind of have to sit with it for a little bit after class, and then it will
definitely.
make a little bit more sense. All right, so acetone. What does acetone do? Acetone is actually the simplest ketone. It's just that. Okay. And if we take acetone and we put it with water, we can then come up with a mechanism. Actually, I think this would be hydronium, right? We have to
acidic environment.
We have to make it acidic, and we have to have a hydronium so that we can readily take off that hydrogen. Okay. And so we have this oxygen is going to do this, right? And that's going to come there. And then we react it with water again. And that's going to come underneath. This is going to be our oxygen-based nucleophile.
Okay.
So, like that. And we have this situation. And put in water again, and it's going to take that hydrogen off.
Yeah.
And so we end up with a, now this is a diol,
Okay. Yeah.
right?
Yeah.
So we are really, so if you take acetone and water, you get a diol.
Diols.
And that's pretty easy to do, right? I mean,
Yeah.
we have acetone in the lab.
Mhm.
You just basically put some water in there and you'll get this.
So is that something, if you're like trying to store acetone and preserve it, you want to like definitely keep it away from water?
Yeah. Yeah, if you have a really, if you live in a very humid environment,
Mhm.
then there's, you know, if you don't have a cap on your
Mhm.
fingernail polish remover.
Oh yeah.
And it also depends on the amount, right?
Mhm.
Like, if a little bit of water gets in there, it'll convert a little bit of it to diol,
Mhm.
but you'll still have a predominant
Yeah.
portion of it be your acetone still.
Okay.
So. All right. So I guess, write out the reaction,
Mhm.
and then write out the mechanism. I kind of just did the mechanism, but the reaction would be
Mhm.
our acetone plus water gives us the diol. Okay, cool. Can I erase?
Mhm. Got it.
All right, so moving on, we can take alcohol and use it as the nucleophile instead of water.
Okay.
Okay, and if we do that, then we make things called hemiacetals or acetals. Acetal sounds kind of weird, but if we call it acetyl, it would be confusing,
Yeah.
right? So acetal has essentially two ethers on it. A hemi meaning half, right?
Mhm.
Hemiacetal has one ether and one alcohol.
Oh, okay.
Okay. And so formaldehyde can be turned into a hemiacetal, which is formalin. And that's what we use as a fixative for the most part. So if we had formalin, I think formaldehyde is that. And if we react it with, in this case, an alcohol, like ethanol, then you see what's happening here? We're going to take off this group, or take off this hydrogen here, and we're going to tack it on here.
Okay.
Okay, so
it
this we take this whole group
Uh-huh.
and we put it on to that.
So if the oxygen's charged, it becomes a nucleophile? Is that what's going on?
Yes. Again, these are all oxygen. It's up there.
It's up there, yeah.
It's hiding. It becomes the nucleophile, and then so if we were to then continue to subject it to alcohol, right? Then minus H2O, that means it's a dehydration reaction.
Mhm.
H2O essentially leaves,
Mm-hmm.
and we get the acetal. Okay.
Kind of a side note, but the formaldehyde, you said it's
Yeah.
like a preservative. Like biologically, how does that like preserve things?
So it forms a cross link
Hmm.
on the DNA molecules and the protein molecules,
Mhm.
predominantly. I don't know about lipids. I don't think lipids. It's primarily DNA and protein.
Okay.
And so, because they are now crossed, it forms like this bridge that connects them all,
Hmm.
so they can't biodegrade.
Oh, okay.
So that's how the formalin formaldehyde does that. It binds chemically to the nucleotides from DNA or the amino acids, proteins. And so, all of these polymers have a certain shelf life. DNA sticks around for a long time. But what facilitates that breakdown are going to be things like microbes.
Okay. Yeah.
And so the microbes can speed up breaking down proteins and DNA, but if it's now cross-linked with formaldehyde, or we'll draw out formalin, the microbes can no longer break that stuff down. Okay, their substrate, or the enzymes that they use for that substrate, won't bind there anymore. I can't recognize it because it has these molecules sticking them together, and so it's an unrecognizable structure, and so it doesn't break down.
That's interesting.
All right, so what do we do here? We have what are we going to attach on here?
Oxygen.
Oxygen, right? So the oxygen, and then it's going to be an ethyl group like that, right? And then this is going to become
protonated.
And so this is essentially the hemiacetal, and that's formalin. Okay. Formalin will work, but if we then converted that to an acetal, right? So, this would be then that isn't going to have the same ability to cross-link.
Okay.
It's too far removed from that structure. Okay.
So is that equilibrium? Like it can go back and forth between the acetal and the hemiacetal? Okay.
Yes. And that's important for a number of biological processes, because that's how sugars are made. Sugars have ketones, and we can turn those into hemiacetals or acetals, but when we make the polymers of sugar, so a complex carbohydrate or a sugar ring, I'm sure when you're doing, maybe it was a Fisher projections, where you're looking at different sugars to see is the OH group on this side, or is it on this side? Sugars can be linear, or most times they're in a ring, right? But we need to be able to utilize them to make ATP, and so being able to convert back and forth is important.
Okay.
We'll also talk about how we can use these hemiacetals as protective groups to actually prevent further reactions to occur, and then we can just convert them back to a ketone.
Okay.
So preventing oxidation. And so, yes, having this equilibrium, the ability to go back and forth is handy. We actually exploit that biologically and in the lab.
So you can use that to regulate the metabolic process, or with equilibrium?
Or, well, in terms of biochemically, we use that to be the difference between storing sugar in the form of glycogen, and then breaking that down and utilizing it to make ATP.
It's cool. I know this concepts are familiar, but to see like it molecularly is different. It's interesting.
Right. Yeah. In biology, you just learn glycogen breaks down into glucose, and then glucose. You might, in biology, they may go a little bit into the Krebs cycle and stuff like that, glycolysis, but yeah, you don't see it quite at this level.
Yeah.
And a lot of times in OChem, you don't see this in applicable scales, right?
Yeah.
You just see it like this,
Right.
and you don't see the relevance behind it.
Yeah,
right?
yeah, exactly.
So
It's most interesting when they overlap. Yeah.
when you can try and overlap it, it makes it. I think it makes it stick more.
Mhm.
All right. So here is that mechanism. So right here we're making our hemiacetal, and if we continue, we make our acetal further. All right.
Mhm.
And essentially it's going to be nucleophilic addition. So this whole process here of the mechanism is nucleophilic addition, right?
Mhm.
We're using this alcohol as a nucleophile to add it on to our ketone, right? And then we have loss of a leaving group, all right? What we've done here is we have tacked on water, or we've essentially not tacked on water, but we've tacked on we've done a proton transfer to make that a water molecule, which is then
Good.
turning into a good leaving group, right? So this part here is going to be nucleophilic substitution, right?
Mhm.
Because we turn that into a leaving group and we substitute that position with another alcohol.
Okay.
Make sense?
Yeah.
So nucleophilic addition followed up with a substitution. So if we stop at the addition step,
Mhm.
hemiacetal.
Okay.
One alcohol group, one ether group. If we continue it further and we do that substitution,
Mhm.
now we have an acetal with two ether groups.
And so, what is like the like fork in the road that determines whether it stops there or keeps going? It's having the
How much alcohol you have, the
Okay.
ratio,
The ratio.
your molar ratio.
Oh, okay. All right.
Okay. Yeah.
Cool.
So you have to titrate that just right to where
Mm.
it will replace. So if you have a 1-to-1,
Mhm. It'll stop.
it should probably replace every molecule with one ether group, right?
Mhm.
If it's a 2-to-1
Mhm.
or in excess,
Access. Yeah.
then you drive it to an acetal.
Okay.
Good question. All right, let's take a break.
Cool.
It's almost been an hour, so we'll take a break, and then what I want